All right, welcome back, everyone. So, we just got finished talking about passive transport, and for all the ways in which we describe material entering the cell, whether it be through simple diffusion or facilitated diffusion, the cell didn't have to expend any energy or the cell dollar ATP. Now, let's get into the movement of materials into or out of the cell that will require the use of ATP. This is called active transport. This often occurs with the help of proteins, and it can include moving materials against their concentration gradient. So if you remember, in other words, that would mean moving the materials from an area of low concentration to an area of high concentration. So anytime you do that, you're going to need to use ATP. You're going to need to use energy because normally materials would like to move from an area of high concentration to low concentration. So a lot of times active transport involves proteins that serve as pumps, and that word pretty much accurately describes what it's doing. ATP is required for these membrane proteins to transport substances from across the membrane into the cell. One really important example of this is the sodium potassium pump. You might also see it written as the Na plus slash K plus pump because those are the terms to use for those elements. Sodium is Na and potassium is K. Notice that they both have pluses, meaning that these are the ions that we're pumping across. This pump will transport sodium out of the cell and brings potassium into the cell. It's embedded in the membranes of many cells, but they're particularly common in nerve cells because nerve cells need to maintain an electrical gradient between the inside and outside of the cell. The pump, what it does is it moves 3 sodium molecules out of the cell and brings 2 potassium molecules into the cell. So you can see you're taking away 3 positive charges and bringing in 2 positive charges, and that keeps the gradient around. Cells also get sort of creative with how they use the work done by active transport. So one example is that the sodium potassium pump maintains this high concentration of sodium ions outside of the cell. Therefore, if the cell needs sodium ions, all it has to do is open up a passive sodium channel. And due to this concentration gradient of high sodium concentration outside and low inside, the sodium ions will just diffuse into the cell passively. So it's kind of like the cell uses the active work of the sodium potassium pump to then benefit from not having to use energy to get sodium back into the cell when they need it. And so here's a diagram of the sodium potassium pump, and you can see that in the outside of the cell, we're getting ATP used to pump out extra sodiums and bring in potassiums, okay? You can also see the use of one of these high energy bonds breaking and going from ATP to ADP. All right. Another creative way that cells utilize their active transport energy is through symporters. Symporters means like importing at the same time. So, for example, this sodium glucose symporter uses the sodium ions to pull glucose molecules into the cell. So, because cells will store glucose for their energy, glucose is typically at a higher concentration inside of the cell than outside, meaning that glucose just won't passively come into the cell. However, due to the action of the sodium potassium pump and the fact that sodium ions are now going to easily diffuse into the cell because of their concentration gradient, when the symporter is opened, the flood of sodium ions through the symporter provides enough energy that it'll allow glucose to move through the symporter and into the cell at the same time. So it uses, again, this active transport. The sodium potassium pump creates this ion gradient. It opens it back up, and at the same time, glucose can come into the cell. So it's kind of just getting creative with conserving the energy that it uses. Other types of active transport aren't going to require proteins to help. One example is endocytosis, and endocytosis is the act of bringing materials into the cell, either by enveloping it through phagocytosis or by pinocytosis. So enveloping it in a portion of its cell membrane and then pinching off that portion of membrane. So the first one is phagocytosis, or cell eating. It is when a cell engulfs large particles and then a vesicle containing the particles is pinched off, and then the cell can do what it needs to do with these materials, perhaps digest it or take it somewhere else to the cell. In contrast to phagocytosis, Pinocytosis is called cell drinking. And all that basically, the difference here is that instead of like engulfing a large physical particle, it'll just bring in some extracellular fluid, like it was kind of, I don't know, sipping it through a straw, and then you'll have extracellular fluid with maybe whatever materials it needs in it. And so it's kind of the same idea, but instead of engulfing it, they're kind of just sucking it in through a straw, I guess, and that's kind of how I think about it. Um, so what cells actually do phagocytosis? One example is immune cells. They'll phagocytize invading pathogens. And what they'll do is they'll phagocytize it, they'll bring it in, and then they'll dismantle it, degrade it, or digest it, basically, in that vacuole. Um, when cells do endocytosis, they aren't always very selective. You can imagine that it's pretty hard to be selective. If you want this thing, but maybe you don't want this thing, well, if you open up, there's a chance that thing's coming with it. But they actually can be selective if they use receptors on the outside of the cell. And if they do that, that's called receptor-mediated endocytosis. Basically, endocytosis being mediated by receptors. They use the receptors on the outside of the cell, and it'll bind to a specific material. And once the receptors have been bound to enough of that material, then they will undergo phagocytosis. And one specific example of that is red blood cells. Red blood cells will use the receptors on the outside of their cell to bind to iron. And once they have enough of the iron, then they will phagocytize that iron. So there are ways for it to be a little bit more selective. In contrast with endocytosis, there's also exocytosis. Exocytosis is simply the process of a cell exporting material using a vesicle. So there's material that the cell makes that it needs to get out of the cell. Either it's something the cell has built and or produced that needs to get out to the rest of the body, or maybe it's just something the cell needs out because it's toxic. It will produce these materials and form a vesicle around it. That vesicle will combine with the plasma membrane, release the materials into the extracellular fluid, and then that vesicle will just become part of the cell membrane. It's made out of the same material, the phospholipids. And so it's really awesome how that can work, how you can just take a vesicle, put it out, and you don't have to worry about these materials anymore because they'll just become part of the membrane itself. So I think I'm going to stop that here. It's a rather short one. And next time I see you, we will talk about section 3.2. Yeah.